All right. Hello, everybody. I'm Ethan Bass. I'm just defended my PhD in the Kessler Lab at Cornell University, and I'm going to present a chunk of my dissertation today. And I'd like to thank some of the organizations that have funded this work, CMID and the Atkinson Center for Sustainability. And I'd also like to thank my wonderful undergrad, Anna Goodman, who helped out with some of this work, and also Nuri, you helped with some of the work. Thanks for coming. Way back. So I want to take us for a minute to an old field in Ithaca, New York, to set the stage for my talk. So these are early successional plant communities that tend to develop very rapidly on abandoned agricultural fields and on the sides of roads and things all across the Northeast and the Midwest of North America. And they're really conspicuously dominated by one of my favorite species of plants that I'll be talking about today, tall goldenrod, which you can see growing all over this photograph. And this has been a really fruitful system for investigating the effects of biotic interactions on community assembly. So a lot of this work has focused on the role of herbivores due to the large and diverse community of insects that feed on these plants. So I see some ball gall folks out there in the audience. And so this is one of the first systems where insect herbivores were directly shown to influence plant community structure. So this is a famous experiment conducted by Carson and Root, where they experimentally excluded herbivory using insecticide. So on the left here, you can see a plot with natural herbivory represented by this gigantic Chirabda leaf beetle. And on the right, you can see the sprayed plot where herbivores were excluded. And what they found is that goldenrod density and abundance increases dramatically when you spray these plots, suggesting that the dominant herbivores in these communities, like these leaf beetles, can reshape the community by suppressing the dominance of goldenrod. And given these large effects on community structure, you might also expect to see effects on the population structure of our dominant species through natural selection. And that's what we find. So if you take plants from these two populations and grow them in a common garden, you find divergence in a number of traits. So plants from the sprayed population have reduced resistance to herbivory and also increased competitive ability. And in addition, we also see pretty large changes in the secondary metabolite profiles of these plants, so chemical defenses. And in general, these defenses are reduced in the insecticide-treated plots, as you might expect. But one major exception to this is the class of compounds I want to talk about today. So these are the polyacetylenes, which are only produced in below-ground tissues of goldenrod, interestingly, so in the roots and rhizomes. And so in the common garden, you see a large increase in the concentrations of these compounds in plants sourced from the insecticide-treated plots. So it seems like herbivores drive selection on the expression of this trait. And we also see a very similar pattern, sorry for the blurry image. You also see a similar pattern in the invasive range. So these are three populations from the southeastern United States. And you can see that they're all fairly similar in their polyacetylene content. And if we compare those, what happened here? This is the wrong one. If you compare those to the population from Japan, they're much higher. Somehow this is SLA and not polyacetylene content, so you'll have to take my word for it. Sorry about that, but this is a great paper that just came out if you want to look it up for yourself. And this has also been shown in a number of other studies previously. So these polyacetylines are also under strong selection in the invasive range. And when you grow different goldenrod genotypes in a common garden, polyacetylene content tends to be really strongly associated with goldenrod performance. So this is DME content on the x-axis and above ground biomass on the y-axis, and plants collected from a population in Ithaca. You can see that these are pretty tightly associated, suggesting that these polyacetylines increase goldenrod fitness. And we see the same thing in plants sourced from across the United States and also in populations from Japan in the invasive range. And we see a very similar relationship between DME and other fitness proxies like inflorescence mass and rhizome mass, which is a proxy for vegetative reproduction. So what drives this strong relationship between DME content and goldenrod performance? What do these compounds actually do for the plant? So we know that the distribution of this trait is shaped by herbivory, but we also know that these compounds are only expressed in roots. So it's not really clear how leaf beetles, which feed on leaves, are driving selection on root secondary metabolism. And one hypothesis that has been studied a lot is that these compounds play a role in allelopathy. So it's known from lab experiments mostly that polyacetylines strongly inhibit the germination and growth of seedlings. So this could increase goldenrod performance through suppressing the growth of heterospecific competitors. But we also know that these compounds have really strong antimicrobial activity. So they may function through the suppression of pathogens and promote increased performance of goldenrod that way. So I'll be focusing on this second hypothesis today, but I'm happy to talk about the allelopathy. So I wanted to test whether selection on polyacetylines is mediated by microbial interactions. And if this is true, we'd expect that polyacetylines increase goldenrod performance by suppressing pathogen growth. And selection on polyacetylines may also vary across different microbial environments. So to test this, I set up a common garden experiment with different genotypes of goldenrod, all from like a local population in Ithaca. And they vary genetically in their polyacetylene content. And I manipulated the soil microbial community using inocula from our long-term succession experiment, also in Ithaca. So this is just a grid of plots that are maintained at different stages of succession. And we know from prior work that the microbial community changes over succession. So what I did is I took soils from early succession, year one, after agricultural abandonment, and late succession, so after 6 years of development, and inoculated small volumes of soil from those plots into these different goldenrod genotypes. And there's also a sterilized control inoculum. So what I found is that the benefits of polyacetylines do shift over succession. So this is just a scatter plot with DME again on the x-axis. So this is the major polyacetylene in goldenrod roots, and above ground biomass on the y-axis as a proxy for fitness. So I'll show you the results first from the sterilized microbial background. And so in this environment, there's no real effect of polyacetylines on goldenrod growth. There's a slight negative slope, but it's not significant. And similarly, in the early successional background, we get a flat line so no effect on goldenrod growth. But in the late successional microbial community, we see a positive slope indicating that polyacetylenes increase goldenrod performance only in this late successional microbial background. And if you compare across these three lines, you can see that the low polyacetylene genotypes are pretty strongly inhibited by both of the live soil inocula, suggesting that they're experiencing some antagonistic microbial interactions. But on the right side of the figure, those lines cross, suggesting that the high polyacetylene genotypes are resistant to these microbial effects. So overall, this suggests that polyacetylenes do enhance goldenrod performance through a microbially mediated pathway, probably through the suppression of pathogens. And selection on polyacetylenes does vary across microbial environments. So in pretty much any population that you look at, we see pretty large variation in polyacetylene content. And these polyacetylines are also highly heritable. So I think the question naturally arises, what maintains this variation in polyacetylene content that we see within populations? And so there could be an energetic cost to polyacetylene production, perhaps, but there could also be ecological costs arising from the inhibition of beneficial microbes. So I was interested in looking at the effects of these polyacetylenes on mycorrhizae, because we think these are important for nutrient acquisition in this system. And so I did another common garden experiment to look at this with like the same, so different genotypes varying in polyacetylines, and they're inoculated with spores from the model AMF species, Rhizophagus irregularis. And so this is another scatter plot, DME on the x-axis and mycorrhizal colonization on the y-axis. And you can see that genotypes with higher polyacetylene production have reduced colonization. And since this is just a correlation, I also looked at this in an in vitro experiment, so with a crude extract and a purified DME extract. So here you can see that the crude extract containing polyacetylenes, along with other secondary metabolites from the roots, suppresses hyphal elongation, suppresses performance of the mycorrhizae, and we see like an identical pattern with the DME extract, suggesting that DME completely accounts for the suppression that we see in the crude extract. So skip. And so it seems that the variation we see in polyacetylene investment may be maintained through balancing selection. So you have all these different guilds of organisms that are interacting with goldenrod. We have herbivores, we have pathogens, we have mutualists, and I showed you that the herbivores are definitely driving selection on polyacetylene content. When you wipe them out, you get increased polyacetylenes. And I think there's a lot of interesting potential here for eco-evolutionary interactions, feedbacks between these different guilds. So thank you very much.
So social life comes in many forms, from loose and uncommitted interactions to highly interdependent cooperators. And I'm really excited about how this happens. And so how does evolution build not just cooperation, but commitment? Not just sharing, but obligate interreliance. And so that's the reason I'm interested in these behaviors. These are called social transfers. And these behaviors transmit endogenously produced materials, often gene products, between individuals. And these socially transferred materials that are transferred, things like milk or egg or ejaculate, are passed between conspecifics, and they bring about a direct physiological response in receivers, typically bypassing sensory organs. So things like pheromones don't count. And to me, the really exciting part of socially transferred materials is that they aren't just two organisms interacting. This is the physiology of one organism reaching over and manipulating the physiology of another one. And so generally, socially transferred materials are passed in extremely important fitness-relevant behaviors, either in sex and reproduction or in care-related behaviors. And that said, each transfer likely evolved first to transmit one particular thing for a primary cargo. So for example, gametes or nutrition or symbionts. And then as evolution has churned on, other components have gotten added to these primary cargoes to either enable better transfer or to accomplish other, sometimes more manipulative functions. And so social transfers, broadly speaking, have had major evolutionary implications. So new social transfers have resulted in not only new organs, but also whole new secretion systems. Mammalian sweat glands, for example, have transformed to nourish young, and now all extant mammals rely on that social transfer. And so these innovations have led to profound shifts in ecology and evolution. For example, think of lineages that use calcified eggs, or of the great, there's more seats up in front, come on, skip in the back. Or of the great success of placental mammals. And you know, there's a lot of us now, and we're ruining the earth, to show you how important it was. So socially transferred proteins, so things that are transferred in these behaviors, can show strong signatures of selection as well. And the best known example of this is probably in testes, where many different genes that are expressed there show signatures of positive selection and also a high rate of gene birth. In other systems, like my ants, which I'll tell you more about, ants that use social regurgitation, the genes are often duplicated and neo-functionalized as they come to act in the socially transferred material. And again, these often also show clear signs of positive selection. So beyond the classic social transfers, biology has many other fascinating social transfers. So think deep in your hearts about social transfers that you're studying and come talk to me about them later. So social transfers vary in really interesting ways. So for example, if we consider the interaction between relatedness and the rate and duration of the transfer, you can see a link. So generally things that are between partners of low relatedness tend to be brief or one-time or asynchronous transfers, while transfers between partners that are very highly related tend to be more sustained, more frequent, and synchronous. And so this leads me to a topic I think is really exciting, is something found in social transfers of care, which is metabolic division of labor across partners. And so what do I mean by this? So metabolic division of labor allows the decoupling of resource acquisition and use in time, space, and across individuals. Think about the mass of biological reactions that you have to do to produce a new you. There's a lot. And think about all of the energy that is required to run all of those reactions. It's also a lot. Now imagine instead of having to run all of these reactions and make all of those enzymes yourself, you only had to run some because someone else was providing you with an intermediate. And so this way, metabolic division of labor enables greater specialization and greater efficiency. It also tends to entrench the cooperation. So over evolutionary time, offspring, for example, tend to become dependent on parentally provided metabolic shortcuts. So this is a study that I've been working on for a number of years now in collaboration with Becky Kilner's group, where they did an incredible experimental evolution study with populations of burying beetles who can provide who always provide parental care, but sometimes they do it with social transfers and sometimes without. And so they evolved these populations with and without care through social transfers over about 50 generations. And so what we found was that when larvae evolved with care, but then were reared without the care through social transfers, they barely survived, and they were particularly pathetic at taking up food from their diet, which we could see from the proteomics that we did, matching to both the beetle and to the mouse they were eating. And thus we can see the requirement for metabolic division of labor transmitted through a social transfer becoming entrenched, with significant implications for evolution, for resilience, and for ecological success. Okay, now halfway through the talk approximately, I'm going to start telling you about the primary social transfer I study, which is this one, trophallaxis, social regurgitation. About 3/4 of all ants engage in this behavior, and this is basically one ant regurgitating and the other ant drinking it. Some species of ants do this, some don't. And that is in striking contrast to other social transfers like milk, where everybody relies on it. And it makes ants an excellent model clade to study the evolution of social transfers. So social insects actually have many different social transfers that can allow metabolic division of labor to occur. And in species that use social transfers frequently, these fluids that are transferred are rich in proteins, in small RNAs, small molecules, hormones, and more, making this social fluid that's passed around more like a social circulatory system that unites this superorganism than a simple secretion. So when do these social transfer behaviors become more than just a behavior, more than just care, and become an evolutionary force? In particular, when does metabolic division of labor lead to a major evolutionary transition in individuality? So this is why social insects are so great to study. So if we think about superorganismality in ants, what should it look like? We have high cooperation between units, we have low conflict, and there should be strict reproductive division of labor, as in no reproductive autonomy of the helper units. So like the cells in our body, the germline-soma distinction. But that doesn't actually work for all ants. There are actually quite a lot of ants that do not display these superorganismal traits. So for example, the orange-ringed ants, these ladies have fully reproductive workers where they can, the workers can mate, they can lay diploid eggs, they can make a new colony if they want. And typically, when you have this sort of break, this not really superorganismal ants, they have particular dietary habits. For example, dietary habits that do not include metabolic division of labor through social sharing. And on the other hand, the blue-ringed ants, they tend to do a lot of trophallaxis and have very individualized care of their young. So what can we learn from this pattern? So we chose a few traits to investigate that we thought would be linked to the evolution of social regurgitation. Within colony, and I'll tell you those. So within colony reproductive conflict, so worker reproductive autonomy, can the workers make a new queen if they want to? And whether a species evolved to eat sugary liquids, so if you're going to do social regurgitation, it might be an important pressure. And this also takes advantage of the angiosperm terrestrial revolution, which was underway when the ants were evolving. And then lastly, colony size, which has often been linked to social complexity. And so we collected trait data and did ancestral state reconstructions and a lot of things that I don't have time to tell you, but are in a preprint, and other investigations. And to summarize what we found using phylogenetic path analysis, and other things, is that evolution of social regurgitation was inhibited by worker reproductive autonomy. So in a situation where there's more reproductive competition, there's less willingness to share between individuals, and they didn't do it. We also saw that the ecological shift towards a sugary liquid diet, which we call eco-opportunism, promoted trophallaxis, or vice versa. We can't tell the difference. They happen at the same time. And then we also saw that once a lineage had evolved social regurgitation, larger colony sizes evolved. And we further found, and so in summary, they needed a reason to share, like eco-opportunism, and a willingness to share, low conflict, and only then did they evolve this social transfer. And further, we found that once they did evolve that social transfer, it increased net diversification across ants. And we did this using both HISI and BAM. So, a social transfer was born. What were the consequences? How did social transfers lead to a suite of superorganismal traits? So this is a great project from an awesome student, Arthur Matt, about queen worker dimorphism. So queens and workers are hugely different in size in some species and same size in others. So we have huge quantitative variation, and it kind of signals a colony-level social control of development. Somehow the colony decides this one's going to be a queen and this one's going to be a worker. But how does this happen? So Arthur realized that larval feeding habits must be the key. So he built up data sets on what ants are eating, what the adults are eating, on larval feeding habits on the scarce species where it's actually been clearly documented. And then he used the incredible work of George and Jeanette Wheeler, which has been collected over many decades, to assemble a database of larval morphologies. And what we could see here with path analysis, and we also have tested it with Pagel's method, is that once we shift the adult diet away from predation, that leads to a shift from semi-autonomous feeding, where workers bring in the prey and the larvae can go ahead, to directed assisted feeding in personalized interactions. And then this led to morphological shifts in the larvae. And further, to link actually this larval morphology and the dimorphism, we ran PGLS, so phylogenetic least squares, to connect these different traits. And we found a lot of things were associated with dimorphism, but what led to what? And so again, we used phylogenetic path analysis to test a number of causal models. And what we see here is that there are sort of two paths that this can happen. One is a predatory diet leading to larval passiveness, which then leads to dimorphism, and then the interesting traits that are consequential. Or predatory diet leading to colony size changes, which can then feed back towards a queen worker dimorphism. So fundamentally, both things are happening. We find stronger push through larval passiveness, which also leads to changes in colony size. So as takeaways, social transfers allow metabolic division of labor, which can lead to increasing interdependency. Motivation, so eco-opportunism and social conflict, led to the evolution of this new social transfer. And then harnessing metabolic division of labor is really what allowed ants to essentially conquer the world through net diversification, high dimorphism, and their large colony sizes, which lead to ecological dominance. And lastly, if you're interested in social transfers, join the Social Transfer Network, and we'll think about them all together. We are running a lot of interesting things. And with that, I thank my team and my collaborators and my funders and you, and happy to take any questions.
Okay, hello everybody. Although it is almost the very end of our outstanding conference, I hope that the perspectives of my team about cancer and the evolution of metabolic rate is no less important. So, cancer. Cancer remains a major cause of death worldwide, charging its annual toll about 10.1 million of people each year, and poses a severe threat to public health and modern economy. Moreover, its burden is predicted to increase in the next 3 years, what is easily seen in the last 2 decades. So basically, the studies concern the causes, the characteristic of development, and clinical treatment of cancer become the foundation of modern science, and presently expand broadly beyond classic medicine. So the idea of cancer etiology and the mechanism of its development in the field of evolutionary biology got a special interest in the context of so-called Peter's paradox. The assumptions of this paradox are very easy to follow and consist of 2 major points. So first of all, we can assume that every living organism is built of cells. I hope we can all agree about it. Yeah? So, if we can consider body size or body mass, I will use these terms as a synonyms, are basically the sum of number of cells. So the bigger the organism, so the greater number of cells that builds its body. Yeah. With the second point, we can fairly assume that every single physiologically active cells have more or less the same chance, the same probability to get a cancerogenic mutation. So if we mark such a chance with a p, so for single cells it will be just 1p, for two cells it should be 2p, and then for multicellular organisms that are built of n cells, the risk of cancer mutation should be n times higher. Yep. So any evolutionary increase in the number of cells and thus a body mass should lead to higher cancer frequency, and all else should be equal. So the biggest organisms like elephants or whales today should have the biggest chance to getting cancerogenic mutations. Moreover, as bigger animals tend to live much longer than smaller ones, these cells need to divide much more time and increasing the chance of getting cancerogenic mutation with every single cell multiplication. However, available data failed to confirm any correlation between cancer incidence and the body mass or longevity. And the lack of this relationship was dubbed a Peto paradox after the name of Richard Peto, who first formulated this idea. Hence, if Peto paradox described a real phenomenon, the natural selection should play a very important role in the development of cancer, mechanism of cancer resistance, especially in large and long-lived animals. So interest in Peto paradox has increased after it has been postulated that the mechanism that can explain its solution can provide new methods for cancer prevention and treatment. So today, many hypothetical compensatory mechanisms has been proposed, and they may include, for example, slower somatic cell turnover or evolutionary elimination of proto-oncogenes or tumor suppressor genes that may undergo cancerogenic mutations. Or, from the other hand, they may simply increase the number of protective tumor suppressor genes, as for example in case of elephants, which have, for example, 40 copies of protective p53 gene, while humans have only one copy. Another hypothetical mechanism goes to the more efficient immune system, better suppression of inflammation or enhanced resistance for oncogenic viruses or bacteria, and so on. Nevertheless, today many such molecular and organismal adaptations have been proposed that can explain Peto paradox and develop the new methods of cancer prevention. However, any practical considerations for explanation of Peto paradox has made no or just little progress, as this phenomenon hits several important constraints. So one of the cardinal arguments that should be addressed here is literal interpretation of Peto paradox on its key assumption on comparing large versus small animals. Body mass constitutes a basic biological trait, and no single factor is more dominant in constraining animal design. Moreover, it has such an overwhelming influence on the evolution of the organism that comparing small and large animals are fundamentally impossible due to the multitude of the factors. Another point that should be underlined here is that any explanations of Peter paradox through the changes of cancer mechanism suppressions, comparing different species, is just unreasonable because evolution proceeds at the population level, so within species. So species differ with respect to the mechanism of cancer suppressions because they evolve independently of each other. Yeah? And as animals display a dramatically different body masses, any transfer of molecular anti-cancer mechanism between phylogenetically distant species seems just impossible. Another simplification goes to the size of cells, to the invariance of size of cells that build our bodies, and the isometric scaling of body mass, body size with the number of those. Today, it is quite striking that cell size differ at both inter and intraspecific level. In many groups of organisms, here we have mammals and birds. This is a classic correlation, and there are phylogenetic corrected analyses. And we can see that the size of cells depends strongly on body mass, and within the very narrow range of body mass, we can see great variation in size of cells. So, according to the cellular architecture, body mass can consist of large number of small cells or a small number of big cells, or the combination of both, while body mass can remain unaffected. Yeah, so we have the same mass here. Among the risk factor that can produce the cancer, the rate of energy expenditure expressed as a basal metabolic rate may stand for the informative marker of health status and prognosis for most diseases, including cancer development. So higher BMR is generally today related to greater mortality rate and to increased risk of most of cancers due to the excessive production of reactive oxygen species or other metabolic byproducts that can lead to the oxidative stress, or the BMR is also associated with higher cost of maintenance of energetic processes like immune response, or through the more intense turnover of somatic cells, leading to the cellular dysfunction and self-death. And what is interesting, that genetically determined variation in the BMR exists, and its significant part can be attributed to the cell size, which may have a direct impact on many physiological traits and also those related to cancer. So, for example, size of cells is strongly related to the cell division rate. So, bigger cells generally have much faster division rate, much faster cell cycles, increased protein synthesis, leading or promoting mutations, promoting aging, and cancer susceptibility through every single cell multiplication. Moreover, size of cells is also the key determinant of cellular metabolic rate, especially via the cost of maintenance of membrane gradients or mitochondria activity or mitochondria number. And in that case, Bigger cells should have much more lower metabolism. However, in case of metabolically active tissues like, for example, liver or hepatocytes, the multitude of the energetic processes that occur in the cytoplasm of these cells and the needs for the enhanced number of nutritional resources or biomolecules like proteins or DNA or RNA force these cells to be bigger and have higher metabolic rate and of course higher ROS production. That's for the theory, and here we try to test this hypothesis. So we try to test the impact of genetically determined metabolism and variation in cell size on the chance of cancer development and its progression. Yeah? And to do so, we use a specific animal model consists of laboratory mice artificially selected for high and low BMR, and we chemically enforced the liver tumor to see if high BMR individuals developed cancer much more often and much more faster. So I will skip this. I want to show you that the hepatocytes, the liver cells in HBMR line are almost twice as big as in the low BMR individuals. And this have a direct implication in the liver size, so there's a great expansion of liver size in HBMR, BMR line. Okay, I will just show you the last slide, sorry for the timing. I just want to show you that the cancer resistance may occur through the lowering BMR, and that goes through the small metabolic cells, lower ROS production, and, for example, lower cost of immune response. Sorry for the time. If you have any questions, I can answer that after the session.
Hi everyone, I'm Penny and I'm doing my PhD in Sally Otto's lab at UBC. And today I'm going to tell you about some of my work investigating the effect of multiple metal interactions in one of my experiments, evolution experiments using yeast. So I'm a lab empiricist studying mechanisms of stress adaptation in extreme environments. And often in this field, we apply one selection pressure at a time, so we can be confident that any adaptive change we see is associated with that stressor. But it's important to consider that natural populations are subject to very many selection pressures simultaneously, and with anthropogenic pollution, many of these stressors are novel or are being released at novel rates. An example of this is our use and release of heavy metals. And using a system of multiple metals has been really cool because I have a suite of stressors with some overlap in their toxic effects. For example, many metals produce oxidative stress, but each metal also has its own unique features and effects on the cell. So with this system, I'm interested in questions like how do multiple metals interact to affect environmental toxicity, and how does this affect the mechanisms underlying evolutionary rescue? So will evolution in the combination favor adaptation to one metal over the other, or will there be equal adaptation for each? To investigate these questions, I performed evolutionary rescue experiments in all 15 pairwise combinations of 6 metals. And I'm still right in the middle of doing this work, so today I'm mostly going to talk about the metal interactions and just give a first look at some of the fitness data of the evolved lines that I've generated. Okay, so getting into metal interactions. First, I want to present a null model for my expectations of how these interactions might play out. So if there is no interaction between two metals in the environment, then the toxic effect of each metal should combine additively to reduce fitness. An interaction between two metals could cause a deviation from additivity, where the combination of metals is either more or less toxic than expected from the sum of each metal's effect. So when the combination of two metals is more toxic than expected, like in the red box on the right, this is a synergistic interaction. And when the combination is less toxic than expected, this is an antagonistic interaction. So what I did first was take the haploid strain of Saccharomyces cerevisiae, that would be the ancestor in my evolution experiment, and I measured its growth rate in each of the 6 metals listed at the bottom of the slide, and all of their pairwise combinations. And I did this in a gradient of concentrations. So each graph here shows one metal combination, and the growth rates in the single metals are on the outside, and their combination is in the middle. And I will refer to these as V graphs. So if we look more closely, we can see that there is a range of interactions. For example, copper and zinc, shown in the red box at the top, appear to have a synergistic interaction where their combination is super toxic even at low permissive concentrations of each of the single metals. And this ranges up to the example of copper and manganese, shown in green, where their interaction is antagonistic, and the yeast actually grows better when these two metals are in the same environment than either one on its own. And then the rest of these combinations kind of fall somewhere intermediate between these two. So to quantitatively describe the nature of the interaction, we created this scale-free parameter, delta, which normalizes the depth of the V's based on the growth rates in single metals within each concentration. And I calculated these values for each concentration in every metal combination to get a numeric value that describes the strength and direction of the interaction. And here I'm showing those delta values. So I have the metal combinations along the X and the delta value on the Y. And the colored dots represent the different concentrations in each combination, and the black dot is their average. So something that we didn't necessarily expect is that the interaction doesn't change very much with increasing levels of stress. So the colored dots are mostly clustered around the mean. And we can compare this to this cartoon that I drew showing what we would see from the additive model that I presented at the beginning, where the reduction in fitness from max is doubled in the combination. And we can see that delta changes more substantially with concentration compared to the deltas from the real data, maybe with the exception of that copper-manganese combination all the way on the left. So this is a scaleless feature is nice because it suggests that delta is a useful measure to describe the nature of the interaction and can be used to compare interactions between any two stressors at any stress level. And the contributions from each stressor don't need to be identical. And what's really cool is that these combinations can be divided into 4 distinct groups with different types of interactions ranging from strong synergism in group 1 and strong antagonism in group 4. So on the right side, I have example V-graphs from each group to demonstrate kind of their relative shapes. And in the inset, in the graph on the left, I'm showing some idealized examples of how that delta value, which I took from the graph that the inset is in, to show the relative depths of what that would do. And the dashed lines here represent the case when delta equals 0.5, meaning that the growth rate in the combination is half the growth rate in either single metal. So here's the breakdown of the combinations in each group, and I want to point out an interesting pattern here. So all of the combinations in group 1 have copper, and almost all the combinations in groups 3 and 4 have manganese. And I'm going to propose some mechanisms that I found in the literature that might explain this. So manganese is probably protecting the cell from oxidative stress caused by the other metal, because manganese can form these non-proteinaceous complexes with small molecules like phosphates that are actually capable of scavenging reactive oxygen species. Copper, on the other hand, has strong compounding effects with other metals, possibly because it permeabilizes the membrane, which might allow more metal ions to enter the cell. And this may be less strong in the combination with cadmium, because we know that the copper metallothionein chelator, CUP1, it binds copper, but it also can bind cadmium. And then with manganese, maybe allowing more manganese into the cell increases the protective effect of those complexes. Okay, so this whole time I've been talking about the nature of these interactions, and I've shown you that they're highly variable between different metal combinations. And in order to perform evolution experiments in these environments, I wanted to choose concentrations that would both provide equivalent stress from each metal, but also equivalent stress across the different combinations. And I don't have a lot of time right now to go into detail about how I did this, but very basically, I had to calibrate the concentrations based on the tolerance of the ancestor to the combination of metals, so I had to increase concentrations for less toxic antagonistic pairs and decrease them for more toxic synergistic pairs. And here's the design of my evolutionary rescue experiment. And rescue is a very rapid evolution scenario where a population experiences a large change in their environment and either will go extinct unless it has some rare large effect mutation or some allele that is there and adaptive for that environment in the standing variation. And that those cells that are harboring that allele will rescue that population from extinction. So in deep 96 well plates, I inoculated 80 wells with clonal populations of an isogenic strain and exposed them to lethal doses of the 15 different metal combinations. And over the course of 2 weeks, I checked the plates every day to look for growth, and right when I saw the growth, I would sample it to catch those large effect first step mutations. So in this graph, I'm showing a time series of this evolution experiment with the day of the experiment on the X and the number of wells rescued on the Y. And I was able to get a good rate of rescue for 8 of the 15 combinations with at least 1 pair from each group. But for some of the pairs, I was not able to select the right concentrations with the right balance of toxic enough that most populations will go extinct, but not so toxic that there's never any chance for rescue. So that's what the black X's are showing. On the right, I'm showing the growth rates of all of the evolved lines in their evolution environments. And this graph is really messy, but what I want you to take away is that there's a lot of variation in the evolved lines in most of these combinations, suggesting that they have different mutations that are landing them in different areas of a fitness landscape. I've also coded the shape of the point to show the prevalence of the petite phenotype, and petite yeast have lost their mitochondrial function, so they can no longer aspire. Oh, and those are shown as triangles. And we can see a lot of triangles on this graph, so this seems to be a common consequence of evolutionary rescue in these extreme metal environments. So the next steps from here are to assess the fitness of the evolved lines in the single and double metal environments to see how those V-graphs change following evolutionary rescue. And I have over 100 evolved lines, so I'm still in the thick of doing these fitness assays, but for now I'm going to show the results from 5 evolved lines from 2 different environments. So here I'm showing cadmium nickel lines on the left and manganese nickel lines on the right. And each color represents a different evolved line, and the ancestor is shown in black. The skinny panels on the left show growth rates in normal media with no excess metal added, and the larger panels on the right show the growth rates in the same concentrations of the evolution environment. So I'll point out a few interesting patterns again. What we can see is that the growth rates of the evolved lines are different from the ancestor, suggesting that evolution has occurred. And all of the evolved lines are growing slower than the ancestor in the ancestral environment with no excess metal added, suggesting that there are costs to these adaptations. We can see that most evolved lines have approximately symmetric growth in each of the single metals, but the exceptions, like the dark green line on the left and the blue lines on the right, seem to have better adaptation for nickel. So there will be a lot more assessment of these evolved lines coming, including whole genome sequencing to identify the mutations underlying this adaptation. But for now, I'll leave you with some conclusions that I can draw already. So environmental stressors can interact, leading to a total effect that's different than the sum of the parts. Combinations with manganese tend to be less toxic than expected, and those with copper are more toxic. The petite phenotype seems to be a common consequence of rescue in this environment, and all of these metal adaptations come with a fitness cost in the ancestral environment. It seems that most lines seem to be equally adapted to both metal and the environment, while others have more asymmetric growth. So I would like to thank my funding sources, my wonderful supervisor, Sally Otto, my lab, my committee members, and I'd be happy to take questions.
I'm guessing you haven't looked at the data yet, but so I'm asking if you have a prediction. Do you know if you would predict more trade-off in the ancestor growth in the ancestor condition when there's more of the synergistic interaction, or the reverse?
That's a good question. I can tell you that I've also done a similar experiment with just single metals, and what I've seen is that manganese evolved lines tend to have the worst growth in the ancestral environment. So part of me thinks that, but then when the interaction happens, it kind of changes the whole thing. I guess, yeah, I think I would expect a more synergistic interaction to have. I'd have to think about that, but maybe I can get back to you. Yeah?
I was wondering if you have any, hypotheses about why the petite yeast tends to be the outcome in the recipe situation.
Yeah. So it is known that especially cobalt and manganese tend to do a lot of damage to the mitochondria. So it's not necessarily adaptive. It could just be a consequence of being in this environment where those ions are targeting the mitochondria and damaging it a lot. Yeah. Okay. Thank you.
Hi, everyone. My name is Raphael Fougeray. I'm a PhD student at the LASA in French Guiana. And thank you to be here for my presentation. Today I will present you a little part of my studies that focus on the influence of ecological factors on Itomini butterfly biodiversity. So diversity and community assemblages are generally thought to be the result of the interaction between phylogenetic history and natural processes such as migration and stochastic drift, but also abiotic factors and habitat filtering that can limit poorly adapted species, and biotic interactions that can have negative effects such as competition or predation. So the Isomini are a tribe of butterflies that are thought to have originated in the Andes. They are very common, as you can see on the map on the left. They are very common in the neotropics, and they are highly diverse. And they are also widely involved in Mullerian mimicry rings, whereby species share their aposematic signals. In this context, I am interested in understanding what influence Itomini community composition and diversity. Can species richness and assemblages be predicted by environmental conditions alone? And what is the importance of biotic interactions? For this, we conducted our research in Peru and French Guiana with a distribution of sites according to 3 altitude classes in order to better observe variations in communities and ecological factors. So for the rest of my presentation, I will keep this color code. So low altitude in blue, mid altitude in yellow, and high altitude in red. And the choice of 2 distant regions allows for a parallel study where sites are similar in environmental conditions, but differ in evolutionary history. So first, do we find variations in diversity and communities? These rarefaction curves enable us to visualize species richness versus abundance. And overall, we found a significant increase in species richness and abundance with increasing altitude. both in Peru and French Guiana, although overall richness and abundance was highest in Peru. To analyze species composition, we used NMDS plots based on Bray-Curtis distance. On these figures, the sites closest to each other are those that share the most similar species assemblages, both in terms of species presence and abundance. We find sites at similar altitudes are more similar than they are to other sites, and this is true for the both regions. And like for species, we checked for mimicry richness. So mimicry richness increased with the increasing altitude, both in Peru and French Guiana, although this was highest in Peru. And again, like for species assemblages, mimetic communities differed as a function of altitude in both regions. And altitude had a positive effect on both species and mimetic richness and shaped the resulting community assemblages. So since species richness and assemblages was correlated with altitude, we attempted to determine what environmental factors differ with this altitude using world climate data and temperature sensors. Looking at world climate data, we found that both temperature in yellow and precipitation in light blue differed with altitude. Moreover, temperature sensors placed directly at the sampled sites during the sampling periods confirmed significant differences in temperature. So both temperature and rainfall significantly decrease with increasing altitude, which could be limiting factors to ithominy. To disentangle the potential role of these two factors, we replicated our sampling efforts during the rain season when precipitation increase and temperature decrease, irrespective of altitude. We compared 3 sites at different altitudes in French Guiana, sampled both during the dry and the rain season, and found that both diversity and abundance of ithominy increased during the rain season, but only at the lowest altitude, as you can see in blue, where temperatures are highest during the dry season. Temperature is therefore likely more limiting to ithominy than rainfall, and both diversity and abundance may be negatively affected by the higher temperatures in lowland forests during the dry season. In addition to abiotic factors linked to altitude classes, we were interested in biotic interaction and their potential effect on ithominy diversity. As ithominy diversity is positively correlated with altitude, we expect to observe a decrease in predation pressure and competition along the altitudinal gradient. So ithominy is known to have diversified with their Solanaceae host plant, also known as nightshade family. Since diversity of Solanaceae can be ascertained in French Guiana, where the flora is well known, we can use this resource richness as a proxy for potential competition. The more potential host plants there are, the more ithominy species should be able to coexist. Unexpectedly, host plant richness decreased with increasing altitude, suggesting that competition increased with increasing altitude. So although we found an increase in potential resource competition with increasing altitude, we found an increase in ithominy species richness contrary to expectations, suggesting that competition does not reduce species richness. Finally, to measure predation pressure, we placed 200 artificial models of palatable butterflies at each site for 3 days. Although we found a slight increase in predation pressure with increasing altitudes, this was not significant. although this was likely due to the very low predation rates observed at all sites. However, when compared to both species on the left and mimicry richness on the right, we found a positive correlation with increasing predation pressure. Moreover, when we look for the number of species per mimicry rings, we found a positive correlation with the predation rates, which suggests greater convergence in mimicry with increasing predation pressure. Hence, mutualistic interactions may be positively affecting species richness. In conclusion, overall richness was highest in Peru, which makes sense as the tribe originated in the Andes is likely the result of a longer evolutionary history. However, there were positive correlations between richness and assemblages with increasing altitude, suggesting that similar environmental factors are similarly affecting diversity in both Peru and French Guiana. Differences with altitude seems to be the result of decreasing temperatures and not changes in precipitation with increasing altitude. Seasonal changes accompanied by changes in temperature were also shown to affect communities and species distribution. Although increasing altitudes correlated with an increase in resource competition, this was unexpectedly not correlated with species richness. And similarly, unexpectedly, species and mimicry richness both increased with increasing predation pressure, as did mimetic convergence. Mutualistic interactions in the form of mimetic interactions is therefore likely important in explaining increasing diversity despite increasing predation. To continue this study, we plan to take phylogenetic relationships into account in our future community analysis to better ascertain the role of these different biotic interactions in explaining patterns of species diversity and richness. And thank you all. If you have questions. No, okay. Thank you. Yes.
I was wondering, like, how much is known about the host plants of like each of these species, and whether you could look like more specifically, like map them onto the ranges of the host plants or something like that. If that might, but probably not enough is known, right? About what their host
En fait,
plants
pour
are.
le translation, en gros, il demande combien tu sais sur les plantes hôtes et à quel point tu peux corréler la distribution des espèces avec la distribution des plantes hôtes.
For genera is well known, those plants are well known, but for species, there is a lot of species where we don't know the host plant. We know that is in the Solanaceae family, but not the species specifically. And I have seen the work of Kate Sullivan. She do modelization of host plant richness and butterfly richness to compare, and I think I will try it later. So, yeah.
Cool talk, thanks.
Thank you.